In the last lecture, I tried to give you an impression of the range of different soil types as they exist around the globe. And the soils that I didn't talk about are the soils that perhaps are most important for agroecology, and those are the anthropogenic soils. Now, anthropogenic soils take on a variety of forms, to be sure. I'm just going to sort of give you a quick glimpse at two of them. Here is an example of an anthropogenic soil in the Brazilian Amazon. Okay, these are called the terra preta, or the black soils. They were fairly recently discovered, actually. As you know, the European narrative about the Amazon was similar as the European narrative about North America also, that it was a vast wilderness just sitting there waiting for the brilliant Europeans to discover. It turns out that all over the Amazon there are these modified soils. On the left you see a typical oxisol with obviously a lot of iron oxide, or probably a lot of iron oxide given the red color. And then to the right, you see a terra preta, or a black soil. Now, these soils are now well known because of archaeological artifacts that are found inside of them. A variety of other reasons. They're very well known to be anthropogenic. And they probably were created over eons, indicating some sort of a sophisticated knowledge of how soils form and what their possible functions are in agriculture. So there was really intensive agriculture going on here. Intensive in the sense of, probably in a sense of the thought-intensive agriculture using ecological principles. Now, the black color is mainly biochar. Biochar is this material that you get out of organic matter when it's burnt, especially if it's burnt very hot or in the absence of oxygen. So you get these little particles of almost pure carbon. And that does all sorts of things to the soil. It creates a slightly higher pH in the soil. It creates micro habitats for microorganisms to live in, and it has all sorts of potential good benefits to the soil. A lot of research is going on in biochar in general right now, in part stimulated by the fact that we realize that this was such a common technique in the Amazon basin. Here's a map, for example, showing you where not all, but many of the archaeological sites in the Amazon basin are found, and also showing you the extent, that whole area right around in yellow, right around the river itself. Those areas where there's very, very intensive agriculture, indicated by the black soils that archaeologists are finding. So this was actually a technique that was used extensively in the Amazon by the very, very high population density that actually did exist in the Amazon at the time of the European conquest. Unfortunately, all those people are no longer with us, including all of their knowledge, which was lost with the European conquest also. Here's the second example of an anthropogenic soil. This is the Almolonga Valley in Guatemala. There's very intensive vegetable production that goes on here. Here you see a woman with a giant carrot. It's pretty impressive carrot, I guess. And here's a typical traditional way of irrigating. Okay, just with a paddle, you pull out of the canals that extend throughout the valley. You do that. It's a fairly intensive management system. But the most impressive thing about it is the soil management system. Now, the soil management system is quite complicated. It's in preparing their soil, they use a lot of organic material that they gather from the nearby forests. So they take leaf litter from the forest, and they incorporate that leaf litter into the soil when they're preparing their fields. Now, the culture has this I was going to say idiosyncrasy, but it's just a normal part of the culture where there are curanderos, there are shamans. There are shamans that are called soil shamans. And these people know a lot about soils, but in a traditional sense, they know a lot about soils. And so when you as a farmer on your plot, you're having problems with your soil, the vegetables aren't producing, or they're producing more diseases than they should, or something like that, why you call on one of the shamans, and the shaman comes and he examines your soil, sometimes digging a hole and sitting in the hole overnight and getting a good feel for the soil, and gives you a recommendation. And typically the recommendation is various quantities of various kinds of leaf litter. Now, in the surrounding forest, there are basically three kinds of forests. This is an oversimplification, but there are basically three kinds of forests. There are pine forests, there are oak forests, and there's mixed mesophytic forests, you know, the regular broadleaf forests. And so the shaman will, for example, give the recommendation that so many bushels of leaf litter from a pine forest, so many bushels of leaf litter from an oak forest, so many bushels of leaf litter from a mesophytic forest, and you combine that into the soil. Okay, that's the basic structure of the whole thing. And it seems to be very, very old, and it seems to be really very, very efficient at producing high quality soils. Now, one of the most interesting things about the system for me, as an agroecologist, is when you examine the system, here's a slide showing the valley itself, and note the background. The background has the mountains there, and there are mountains surrounding the valley, and the mountains are where the forests are. Those forests are what produces the leaf litter that goes into the agricultural practice. So from the point of view of this culture, their agroecosystem is a much larger thing than just the piece of land that they're working on. As a matter of fact, their agroecosystem includes all of the surrounding forests and all the hills surrounding the actual plot where they're producing their vegetables. It's a vision of an agroecosystem that I happen to think we in the modern world would do well to try and emulate. The agroecosystem isn't just where we put the plants, but it's everything around it also. Should be, and in a sense it is, whether we like it or not. It's a landscape effort. So now let me get on to talking about the details of how these anthropogenic soils work. I'm using anthropogenic soils simply because, for all practical purposes, once you decide you're going to do agriculture, you have decided you're going to create an anthropogenic soil. But there's certain things that happen in soils that you really don't have much control over. So we really need to know what's going on in those soils so we can gain some kind of control. Now, from this, I've shown you this graph several times now, but I try to emphasize the importance here of the soil colloids in the soil structure, important for plant nutrition, which is important for agriculture. In terms of climate change, it's important for carbon storage, of course. But generally, one of the things that we need to understand. Now, there are, the way I think of it at least, there's four basic types of soil colloids. We have the layered silicate clays, we have the hydrous oxides of aluminum and iron, we have amorphic clays, and we have organic colloids. Now, I'm going to talk about all four of those things briefly, but let me begin with actually the most complicated one, and that's the silicate clays. Now, the silicate clays, as you would imagine, is based on a combination of silicon and oxygen. Now, there's sort of two forms that you get these crystal lattice structures out of silicon and oxygen. First of all, you have these triads that form, as in this picture, where the silicon is in the middle, and then you have the oxygen on the outside. And then you get these octahedron formations also. You put and what happens is that's the fundamental form of the crystal, either the tetrahedron or the octahedron, and those tetrahedrons and octahedrons, why then they combine in various ways, using different kinds of chemical bonding, which we don't really have to get into, to form larger structures, and those larger structures are actually the silicate clays. Here's an image from an scanning electron microscope of one type of clay. Now, I want you to get a feeling for this. Of course, they're very tiny particles. They're clay particles, as small as you can get. But you can see they have a lot of structure to them. And you can imagine, remember that all important negative charges that exist on the surface of these things. Well, there's a lot of surface area there that you can see. And that surface area is where all those charges are. Tremendously important for both plant nutrition and preserving the nutrients in the soil so that they don't leach out. Oh, these clay particles, they're formed when the rock, the parent material rock or whatever it is, originally decays chemically over the geological time, and as it decays, and usually this is happening under the surface of the present soil. It's in that rheolithic layer on the surface of the bedrock. And what happens is the elements that come out of it, they combine with elements that are already in the soil solution, etc. And whatever elements are there get incorporated into that crystal lattice structure that I talked about. Okay, so those are the original clay particles, silicate clay particles that get formed. This would be an example, and this is one where you have the octahedral sheet is constrained by two tetrahedral sheets, and then that triplet is connected with another triplet, and that's the basic formation of it's called vermiculite. That's one of the clays that emerges from the original process. Now, during that original process, what happens is as the clay is being formed, as the crystal is being formed, the normal process is you have silicon and oxygen that normally come together like that. But there are other elements, there are other cations that have the same morphological structure as silicon. They're just about the same size, but they have a different valence, a different chemical valence. So they have basically a different charge. And so what can happen, as you see in this particular picture here, what you're going to have is sort of constrained within the crystal structure. You have an element that doesn't really belong. Frequently, that element is aluminum. Frequently, it's iron. There are others also, molybdenum, magnesium. There are others, but the ones that we're mainly concerned with actually are aluminum and iron. But what you can see from this picture right here, from this diagram, is that this is the origin of those surface charges. Okay, because the electrical charge over the entire crystal is sort of averaged from the positive and negative charges in all of the various elements that are together. And if you have on the inside, you have an extra charge, where that extra charge then is reflected on the surface of the clay particle. So this isomorphic, iso meaning the same, morphic meaning structure, so an element with the same structure as silicon, sometimes by mistake, I don't know what that means, but anyway, gets incorporated into the crystal structure, and that creates the negative charge on the surface of the clay particle. Okay, so that's the isomorphic substitution is one of the main ways in which these crystal lattice structures of the silicate clays actually get their their charge. Now, going back to this the little model here of vermiculite, what happens now as the soil, now you have a real soil and it's got its clay particles and everything, and now we go through the process of aging, as I've talked about already. And so from this vermiculite, you maybe get a category of clays that are called smectites, and you can see sort of the structure here. It becomes a little looser. These are the clays that really swell a lot. And then from that structure, you can see easily, I think you can imagine how that might weather into this kind of structure. This is now the kaolinite, which you saw the picture of before. Now finally, the last piece of this is the final decomposition of the tetrahedral and octahedral sheets connected with one another, and you basically get just octahedral sheets that don't really connect with one another very well. They're called amorphous, that is, without very much morphology. Here I have a couple of pictures from electron microscopes, and you can see on the left you have a highly structured clay, and you can imagine with all that structure you're getting all those negative charges, nice negative charges that you're going to use for plant nutrition and everything. And then on the right, what you see is something that we'll call amorphic. That amorphic structure now obviously is going to have some kind of consequences for the kinds of electrical charges that are going to be on the surface of the clay. And now in this image, you can see this is sort of a statement. It's an illustration of the way the charges change. The one on the left, of course, you have a lot of negative charges, as it says in the graph there. The one on the right is a weathered soil with those those amorphic clays inside of it, and you can see what happens there. You have positive charges as well as negative charges. And the other thing is you also have charges that vary depending on what the pH is. Here's an example of an oxisol, and you can see what the cation exchange capacity is for basically the number of negative sites on the clay, negative and positive sites on the clay, as a function of the pH. And you can see the increase in the number of sites as the pH increases. Now we move on to the organic colloids. First of all, let me show you this table here. And in this table, you can see the various charges that exist on different kinds of clays. So we have the smectites and the vermiculites, for example, which have relatively large charges, 100 and 150. Note that 95% of those charges are constant charges. They don't vary with pH very much. About 5% of them are variable charges. Then we have illite. I didn't mention chloride, but it's another kind of clay. And you have a total charge of 30, much less than the smectites. And about 80% of those are constant, and about 20% then are variable, so much more variable. Then you have kaolinite, which is, I talked to you about kaolinite. Kaolinite has a very small charge base, only 5% of which are constant. Mostly they respond to pH. Mostly they're highly variable. And then you have the aluminum and iron oxides, those amorphic clays that are very, very highly weathered. And as you can see, they have low charges, even half the charge of kaolinite. And look at the percentage variable. 100% of the charges are variable. There are no constant charges in these aluminum and iron oxides. Now, this is from a particular soil, okay? I can't remember exactly one. It's from a book, a soils book by Brady. Now, check out the same data for the organic colloids. Obviously, you can see what we're talking about here is the organic colloids almost make it irrelevant what the other soil colloids are doing. I don't really mean irrelevant, but if you have a lot of organic colloids, make a huge difference. They have a huge number of charges. Those charges result in very, very high cation exchange capacity, etc. Now that brings us back to this graph that I keep showing. And what you'll see here now is we really need to add something to this graph, right? Because we have the effect that the ions are absorbed onto the colloidal particles, but now we have these organic colloids. Where do they come from? Well, that's another product of the decomposition. As the soil matter decomposes, the soil organic matter, some of it actually is the colloidal material that acts on this particular piece of the graph. Now, so what we have then are we have two pieces of the decomposition process, and those two pieces of the decomposition process act in different ways. One is the decomposition of the soil organic matter to give ions to the plants to be absorbed by the plants. The other is the decomposition to the point where they're organic colloids that have negative charges on them that engage in cation exchange capacity, and basically making the soil healthy in the sense that it can maintain some of the ions into the system without having them leach out. Now, there has been historically an idea that this is there's certain kinds of organic matter based on its chemistry turns into these permanent soil colloids, while other based on their chemistry turns into the stuff that decomposes to create the ions that the plants can utilize. And these were frequently referred to as recalcitrant or decomposable ions. Those two categories. Now, this paper came out a few years back, and this paper basically challenged that entire idea. Challenged that idea in that it looked at the formation of soil organic matter as a process, an ecosystem process where soil organic matter is constantly forming and degrading and forming and degrading, And it goes right along with this picture that I presented up here right now, that I just presented up here. But this particular paper emphasizes the dynamic nature of the soil properties. And let me read a piece from that paper. Globally, soil organic matter contains more than 3 times as much carbon as either the atmosphere or terrestrial vegetation. I mean, consider that. Let me read that sentence again. Globally, soil organic matter contains more than 3 times as much carbon as either the atmosphere or terrestrial vegetation. A lot of carbon in the soil, in the organic matter. Yet it remains largely unknown why some soil organic matter persists for millennia, whereas other soil organic matter decomposes readily. And this limits our ability to predict how soils will respond to climate change. Recent analytical and experimental advances have demonstrated that molecular structure alone does not control soil organic matter stability. In fact, environmental and biological controls predominate. This is a statement of the importance of ecology in this whole dynamic of the colloidal structure in the soil. So now let us speak a little bit about the organisms that exist in the soil. Here's a bunch of images showing you sort of the various categories that exist in the soil. There really are a lot of things. Sort of functional groups can be pretty easily recognized. For example, here you have things that are basically decomposers. They eat dead tissue. On the other hand, what you have here, you have things that are effectively herbivores. These are the root grazers. These are things that eat that scrape on the surfaces of the roots, and they're actually are herbivores, basically. These things here are obviously decomposers. Well, decomposers and the mycorrhizal fungi that's there, which we talked about earlier, but fungi and bacteria. And there you have it. Now, the 2 things that I didn't mention were the beetles and the centipedes. I mean, beetles are such a huge group, they pretty much can do anything. You have some beetles that are herbivores on the root system, some beetles that are predators, some beetles that eat mainly just decaying and rotting tissue. Centipedes are, for the most part, they're nothing but predators in the system. Now, I really find this graph useful because it's a pyramid of numbers, basically. A pyramid of numbers is one of those things that's pretty constant in ecology. If you look at sort of the size of animals and the numbers at various sizes, they form a particular pattern. And here this illustrates for 1 square meter of soil what you can find. You'll find 1 vertebrate symbolized there by a bird. It could be something else, of course. And as you go down the size scale, you get down to the bacteria and the single cell fungi, which have just enormous numbers of individuals in that 1 square meter. Let me tell you about one particular study now that brings out some of the interesting dynamics that we know exist, but this study sort of laid it out in a great amount of detail. It used this flower. This is a type of flower. It's called Heliconia imbricata in Costa Rica. And a scientist by the name of Rick Seifert decided he wanted to look at the organisms that exist inside of the bracts. Now, those red structures are bracts, and the bracts contain sort of little cups of water, and inside of those cups of water are the flowers. There's hummingbird pollinated, and the hummingbirds come there. But then the flower material, the petals and the stems and the corolla and all that sort of just is there, and it begins decomposing. Now, how does it decompose? And basically what Rick did was he took when the flower was just coming out, it's just a little bud like this, and so you can sort of put a very fine net over the top of it so that organisms cannot get in. The things that would lay eggs there and things like that, they're just not in there. And so what you can do then, when it opens, you can systematically put in whatever you want to put in that little container of water where the floral material is going to be decaying. And you can ask what happens with different combinations of things. Now, as a matter of fact, in terms of the the detritivores, there are basically just four kinds. I'm not going to tell you what they all are because I want to focus on just 2 of them. Okay, you can see them in this blow up here. There's one that's basically a shredder, that it has mandibles that tears up leaf material, and as it's eating the basically eating the surface of the leaf. It's getting all the bacteria and fungi that happen to be there in its mouth, but it's actually shredding the leaf as it goes. The other one, the one with that breathing tube sticking out of the water right there, is a syrphid. Now the syrphid is what I call a slurper. It's eating the decaying material, the material that's sort of liquid and semi-liquid, filled with the bacteria. It's eating. And what Rick discovered, after putting, some flowers he had just syrphids, some just stratiomyids, some had combinations of various different types, and he found a very strong relationship between the syrphid and the stratiomyid. Now at first, he was thinking that this would be a competitive relationship, and that when they were together, they would do worse than when they were alone. The absolute opposite happened. What happened was a very positive relationship between these two species. And in retrospect, we can see exactly how that happened, because the shredder is in there shredding that material. So this stratiomyid larva is shredding the floral tissue. As it's shredding the floral tissue, it's creating microhabitats for the bacteria and the fungi to grow in, forming sort of the bacterial solution, which now is going to be very rich because of all the shredded material, and the syrphid slurps up all that juice. So there's really a very strong and evident positive relationship between the stratiomyid and the surface. Really an excellent study, and other studies could be cited, but I like this particular one because it has a system sort of isolated and contained, but a natural system, so you can see specifically the role of two particular actors in this process. Now let's specifically talk about some complications associated with the decomposers. We talked about the detritivores in the previous section, so let's talk about some of the decomposers. Now the decomposers are mainly your fungi and bacteria in this particular series of images right here. But now we're talking about complications, ecological complications that become quite intricate and not exceedingly well understood for most processes that are happening. Now, for some processes, especially some that are important to agriculture, we do understand them pretty well. I'm going to use one particular example to convince you that it's a complex system, but we understand a certain amount about it, such that we can probably make some rules about the way particular agroecosystems should be managed and should be structured. So I'm going to talk about in particular the nitrogen cycle. Now, at its simplest form, it's pretty obvious. Plant material dies, falls to the surface of the soil, and then the detritivores work on it. They eat it up and they defecate it out, and they process it so that the bacteria can take over. And then the bacteria decompose it so that it becomes soil organic matter, which eventually decomposes to become mineralized. And when the first product of mineralization is ammonium. Ammonium can be taken up by plants, but there's problems with that. But the ammonium then goes on to become nitrate. The nitrate is an ion that gets taken up by the plants also, and that's basically the way it works. And some of that nitrogen is immobilized by the various organisms that are involved in it, as I talked about earlier. Now, completing this picture, we have two things that happen. The nitrate falls out of the system. The nitrate leaches out of the system very easily because it's an anion, and with a negative charge, it usually is not held on the surfaces of soil colloids. Now, it can be held on the surface of colloids when the colloids have positive charges, and some of them do, but that's not really a big thing in most soils. The anions, on the other hand, the anions are held on the soil colloids if the soil colloids are there. If they're not, they can leach out of the system also. There's an additional factor, which turns out to be pretty important from the nitrate. The nitrates then can be denitrified to form nitrous oxide, which, as you probably know, is a very, very potent greenhouse gas. So this process also contributes to the whole problem of greenhouse gases in the atmosphere. Now, at each stage of this operation, there are particular species, particular classes of bacteria that are involved. And I've illustrated here with these little stars, I've illustrated the 6 main places that very, very specific bacteria are involved. For example, the bacteria that catalyze the reactions that change nitrate into nitrous oxide are called, not surprisingly, denitrifying bacteria. Then you have decomposing bacteria, you have mineralizing bacteria. So the species of bacteria fall in these categories. The categories have very specific functions within the nitrogen cycle. So I think that pretty much does it for this lecture. I've gone through quite a bit of material relatively rapidly, but this is kind of the organic part of the soils. This is talking about the organic colloids and emphasizing quite a bit the organic matter and how the organic matter functions. And so that's basically it for this lecture.